All right, Dr. Jussuf Kaifi has devoted his translational cancer research on developing preclinical cancer models with the aim of advancing laboratory findings into clinical trials for lung cancer patients. He's recently made inroads into a new technique to isolate tumor cells in the bloodstream. Now, while he's not working on tumor cells, he can be found sitting at a trap set, playing the drums. And he's really into funk rhythm drumming. Now, if you don't know what funk rhythm is, it's a syncopated style that emphasizes the offbeat. And the drummers in that, you need to think complicated, complex, being focused. Sounds a lot like isolating tumor cells in the bloodstream to me.
Thank you for the kind introduction. Full disclosure, since I play the drums, I don't hear well, so I tend to yell at every turn, so please raise your hands, okay? But Dr. Anna, thank you for inviting me. I feel very honored to present here. I am a general thoracic non-cardiac surgeon, and I treat mostly lung cancer. And my lab studies lung cancer progression, and my lab is at NextGen in Columbia. And I want to start with the unsolved problem in my practice. This is a patient, 64 years old. We treated him in 2021. He had a localized small lung cancer in the right upper lobe that was stage 1, so earlier stage. And he was staged with standard of care imaging, whole body PET scan, brain MRI, no signs of cancer. We did surgery on him, resected the cancer. By the time of surgery, we took a piece of the tissue and grew an organoid in vitro in our lab. And that organoid already showed carboplatin, which is the standard drug, carboplatin resistant. The patient then received treatment, standard of care treatment, adjuvant chemotherapy, also including carboplatin, and he already developed a local regional resistance during treatment, devastating, which we kind of predicted from the lab findings. He then developed a brain metastasis and passed away 16 months later. So the overall problem here is the drug resistance. The drug resistance prevents us from curing lung cancer patients. Lung cancer is by far the #1 cause of cancer-related deaths in America. There are more lung cancer cases than prostate, breast, and pancreatic cancer combined. 5-year survival is 25%, and patients die from recurrences, and the challenge is the drug resistance. In Missouri, we at Alice Fischel Cancer Center, on the center of the state, we treat a rural underserved population. And in the state of Missouri, we're top 5 in terms of smoking rates and of lung cancer rates. And there's a disparity between the rural population in Missouri, which we serve, and the urban population. The rural population does much worse. And when you look at lung cancer-associated survival, you can see that we care of we have the highest lung cancer-associated death rates in entire America. Some of the counties are in the top 10. So really an underserved population in need. When a patient has a localized cancer just in the lung, like the initial patient, when we do blood draw and we look for cancer cells in the blood called circulating tumor cells, we can find them in 60% of the patients already. So at the time of diagnosis of a localized cancer that has no spread on imaging, over half of them have already spreading cancer. And these cancer cells are actually the drug target, because we remove the cancer in the lung, the patients still have cancer cells in the entire body. And these are the targets of the drug treatments. But the problem is these cells are already resistant. And it's very tough to do these studies because you only have very few cancer cells in the blood, just 1 in 10 million. So you can't really do these studies in patients a whole lot. So what we need are models. We need preclinical models that are translatable, that make sense, and that we can use for trials. And we use a library of in vitro and in vivo models. And our lab is now just at the verge where we can translate our findings into clinical trials and then bring that back to the patient. That was always my objective of doing cancer research. I want to bring it back to the patients that I treat. I give you one example of a patient-derived preclinical model that we developed a couple of years ago. Again, it's very tough to grow cancer cells out of the blood. and characterize them. So we had to overcome that. So we took a piece of tissue from a resected lung cancer and grew it in an immunodeficient mouse as a xenograft model. And then we took blood from these mice, just a simple blood draw, and extracted the cancer cells and regrew them in a new mouse. So that way we had a model for the primary tumor and for the spreading cancer. And these models we characterized extensively with single-cell sequencing and all types of technologies, and we identified a metastasis-specific stem cell type and also actionable mechanisms of drug resistance. But doing studies in animals, particularly these xenograft models, is very time-consuming. It takes a long time. It's not clinically applicable. We need high-throughput models with clinical applicability, and one avenue are organoid models. And these work that we take the tumors and we grow a 3D culture, and that's how they look here. We grow a 3D culture of the tumor, and they grow fast. They grow within a week. And when you look at them under a microscope, they look similar to the primary tumor. They maintain the tumor mutations. They have the same transcriptome, and they also maintain the immune system, which is very important now in the era of immunotherapy. We looked at these organoids a little bit more, and what we found, if we found that the organoids from the patients were resistant towards standard of care drugs, and the patients received these standard of care drugs, they develop resistance. They develop recurrences because they were resistant towards the drugs. So what I'm trying to tell is the organoid can predict the treatment outcome of the patient. That's really precision oncology, I think. We looked at these organoids a little more. We did whole transcriptomic bioinformatic analysis on these organoids, and we found an aldoketoreductase overexpressed in the organoids that were resistant. And aldoketoreductases inactivate the chemotherapy. So when a cancer cell has an aldoketoreductase, it inactivates a chemotherapy drug. The chemotherapy does not work. So it made sense. But it got more interesting because we also found a blocker of that aldoketoreductase, which is in clinical care already in Japan and in India for neuropathy. And it's orally available, low toxicity profile, and it has brain penetration, which is very important because lung cancer patients get brain metastasis. And we tested that drug, and that graph basically shows that you can overcome the drug resistance when you add that drug. So the chemotherapy and other agents may work again. And right now we're writing a clinical trial. We are repurposing a pyrostat, which is already tested in humans, and we can advance directly to a phase 2 trial because we know the pharmacokinetics, the pharmacodynamics, and we can now test efficacy in combination with other drugs in lung cancer. So hopefully we can start this trial next year. That's our goal. Most of the findings developed or found in mice don't translate into clinical trials. They're rarely successful in clinical trials, as you all know. I think it's in the booklet as well. And we have the National Swine Center at Mizzou. We have a poster here as well from that center. And pigs are much closer to humans. Physiology, anatomy, immune system, I mean, you name it. And it's great as a surgeon to work with pigs, by the way. I mean, it's fun to work with these great animals. But we can use human-grade instrumentation. We use the same instruments that we use in humans, in fact. But there's a big chance to have more translatability with pigs. And we're investing heavily into this now, our group. The National Swine Center has generated a transgenic, a genetically engineered pig that has cancer mutations that are very similar. They're orthologous to human mutations. And we can induce lung cancer in these pigs locally in any organ site. It's a Krelok system, so you can pick kind of the organ where you want to induce the cancer. We do it in the lung bronchoscopically, and within 4 weeks, the pigs get large cancers. We have a success rate of over 90%, and it's an immunocompetent model. And the pigs actually we house at NextGen. We have a facility now, all the oncopigs are at NextGen in Colombia. And we compared these oncopig lung cancers with comparable mice lung cancers and compared that with human patients, with human lung cancers. And we found that the mouse lung cancers have an overlap of about 30% with human lung cancer, but the pigs are nearly twice as much overlap, 54%. So that's kind of a hint that hopefully this is going to be much more translatable to humans than findings in mice. Yeah, that gets me to the summary. I hope I'm within 10 minutes. So we are studying the major gap, drug resistance. It needs to be a library of preclinical models. Can't just be one, I think, because they're all kind of complementary. They have strengths and weaknesses. And it's all about human translatability and now maintaining the immune system. It's very challenging in these models. And our team at NextGen really believes in multidisciplinary team science. I think it's really about partnership of clinicians and basic scientists, and we have a large group at NextGen that we're really proud of. And I want to thank all of them, and thank you for listening to me today.
Thank you. Okay. Let's have some questions to that. All right, David.
Great talk. It's really exciting to see the progress that you can make as a surgeon scientist, really bridging the clinic to the lab and really scaling all these models. So this may be a little bit hypothetical, but I was wondering if you could tell me what is the current timeline if resources weren't limited to taking a sample, growing an organoid, testing it with all the omic analysis to inform your clinical decision making. Like right now, if resources weren't limited, how long would that take? But then in actuality, how long does it take? What's the gap now where you could really scale this to apply it to do precision interventions for patients currently?
Great question, I think. I mean, the organoids we can grow within 3 days and do drug testing within 6 days. So very fast. Genetic testing, whole transcriptomics, 2 weeks, maybe a little more. But I think the needle ear are really is the availability of drugs. We have many more targets than drugs. And the regulatory restrictions, I'd say.
So are there many scenarios where this kind of testing currently, given those limitations, actually would guide to a different decision in the clinic?
Yeah, absolutely. I think that the 2 cases that we have, I mean, these are small numbers, of course, but the 2 cases that we have, we knew way before they got the treatment that they were drug resistant.
So
but we couldn't tell anybody.
Okay, yeah, so it wasn't really actionable. It was like it informed what you predicted, but it wasn't like you made a decision. So how long do you think it'll be?
We could have changed the decision, I think. They could have gotten different drugs, maybe.
Exactly.
Yeah.
So how long do you think until that happens?
You mean of a timeline when
Predicting
it's going
the future until this is standard of care.
You know, that's a tough question. I would hope within 5 years. I hope that AI may help with this as well, but I hope 5 years, hopefully.
Great, beautiful work.
Thanks.
Mentimeter. And then you can come, yeah.
Yes. Can you describe for us what accounts for the difference in transcripts between the oncopigs and humans?
Say that again. What accounts?
Yes. Can you detail what accounts for the difference in transcripts between oncopigs and humans, if there is a difference in the transcript there?
That's a good question. I don't know. The research, in particular the cancer research, is fairly new in pigs in general, I'd say. So we haven't looked at that. So I don't know. What we did at Mizzou with the life science centers is that we compared the mutations and the genome of pigs with humans and with other species. And pigs are very, very close to humans, surprisingly. The transcripts, the transcriptome, I'd have to look it up. I don't know. Sorry.
All right. Last question.
So the data you show related to the percentage of people that have cancer in rural area versus urban area was a little bit surprising. You would imagine that in the urban area, because we have pollutions, you will have more.
Yes.
So how do you explain the difference in the?
That's a fantastic question. It is in a certain way surprising, but it's the smoking rates. Radon seems to have a rate, and it's their socioeconomic factors as well. In an urban population, I think you balance it out with, you balance the socioeconomic factors out. in rural Midwest is socioeconomically really low. A lot of drug problems, and it's multifactorial for sure, but it's mostly, I think, socioeconomically, and smoking rates are much higher in rural populations as well.
Is it possible that the use of pesticide in agriculture and so forth could have an effect?
Yes, yeah. I think you will find a long list. That's probably part of it.
Thank you.
Good question. Yeah.
All right. Thank you, Jussuf. Let's give him a hand, please.